I'll turn it over to Carla, who will introduce today's speaker.
Okay, well, it's great to have Teresa back at the MVZ. It's hard to believe it's been 11 years since Teresa graduated as an undergraduate from Berkeley. She was one of our first one of our earliest undergrads in the MVZ undergraduate program. She worked on the Grinnell Resurvey project. She did research with Chris Clark, who is a former PhD student here on hummingbirds. And then after she graduated, she spent 2 years working with me on an NSF grant to digitize and catalog our entire audio collection, and which is all online now, thanks to Teresa. And then she went and did a PhD with Rick Prum at Yale, and Rick's coming in 2 weeks, I think, to give a special lecture. And she was there at Yale for 6 years, and then went to the Smithsonian for a research fellowship for 2 years, and you're still a research associate, is that right? And then has spent the last year, ending at the end of this month, doing a science and technology policy fellowship in Sacramento in the state legislature. So she's done lots of different things, published lots of really interesting papers, and she's going to talk to us about what you've been up to since you graduated from Berkeley in 2007. So welcome back.
Great. Well, thanks, Carla, and thanks everyone for having me here today. It's absolutely fantastic to be back home at the MVZ. So I'm going to give a talk today in sort of two parts. I'm going to talk about sort of basically all the research I've been up to, I guess, in the 11 years since I graduated. So it's going to be a little bit of a whirlwind tour of feather research. And then I'm going to end talking about what I've been doing for the last year working in the state legislature as part of this policy fellowship. So we're going to start with feathers. So in general, my research has basically turned into a quest to understand the sort of the relationship between the morphology and function of feathers. Feathers have this incredibly rich diversity of shapes and sizes, and it turns out that birds are doing quite a lot of things with those feathers. So in my last years, my life as a staffer, people ask me for a really succinct, version of what I do, and I say, well, I'm trained as a biologist, I study birds and what they do with their feathers. And people say, great, and then they look at me a little funny and they're like, you mean how they fly, right? Like I'm trying to pull one over. I'm like, well, yeah, birds fly with their feathers, but they do so much more than that. A lot of birds swim with their feathers. We've got multiple independent evolutions of winged propelled diving. Some birds use their feathers to stand out in a crowded lek and get a mate, and other birds use their feathers to camouflage. So last time I gave this talk, I got protests for not pointing out the birds. So here's the tail, or here's the head, and the wings, and the tail. My friend and I almost stepped on that bird. So well lit in. A lot of birds, like ducks, use their feathers to repel water, and sand grouse use their feathers to soak up water and bring drinks of water to their nestlings on the other side of the desert. Owls have specially modified wing feathers to fly silently, and the club-winged manakin has specially modified wing feathers to make what we presume is an attractive buzz during their courtship display. So birds do an incredibly wide range of behaviors with their feathers, and I'm interested in studying these because when we look into it, what they're actually doing with these feathers, we find that underlying all of that is this incredible diversity, right? All of these different shapes and sizes really affect the function that these feathers can perform. So if we take a quick look at the silent owl and the buzzing manakin, the owl has a whole host of structural modifications to their feather. They've got a leading edge comb, a fuzzy fringe, and this trailing edge modifications here that all work in concert to help dampen sound effects as this owl is flying. In contrast, the manakin has a completely different set of modifications to their feather. They've got these grossly swollen central shafts here. This one is bent into a pick. This one has grooves on it, so when the bird snaps its wings together over its back, those feathers rub together and they produce sound through the mechanism of stridulation, right? So really obvious and different things happening with the structure and microstructure of these feathers to achieve these different structures. So that's probably what I'm interested in, this form and function, and I like to study this because we can answer a lot of questions, right? So first is just we'll stand to learn a lot more about avian phenology, right? These birds are doing so many different things with their feathers that if we better understand that form and function relationship, we can, basically learn a whole lot more about just birds in general, which is great for the ornithologist in me. The second is it allows us to get a better sense of the evolution of all of these different features, right? Birds, it turns out, in the last couple of decades, we've just turned up an amazing wealth of fossils of these taxa between dinosaurs and birds that are covered in feathers, right? So feathers predate birds, which means a lot of the behaviors associated with feathers, like flight and display, also probably predate birds. So in looking at the relationship between form and function, if we understand what structural modifications are actually necessary to achieve a particular behavior, then we have hopes of finding that in the fossil record, and we can do a better job of inferring, basically what these feathers were used for in these fossil taxa. And like I said, we've got a lot of great fossils. There's surprising detail in these fossils. We can actually get at the microstructures, particularly in some of this newer material that's coming out preserved in amber, right? We can take a microscope to that and really get at these structures. And the final reason why I like working on this stuff is for the potential to basically inspire new technology. Birds are solving a lot of problems with their feathers that we as people would also like to be able to solve, like flight or silent flight. And in understanding kind of how they're getting at that mechanistically, it can lead to some new discoveries. So here's some examples of some biologists that were looking at how birds make blue. It's not a pigment, but nanostructures inside this feather cells that are coherently scattering light. And this has inspired these folks in material science to develop basically photonic pigments making use of that same mechanism. And they're really excited about this because they think that they'll be less prone to fading than chemical pigments, right? So there's growing number of studies here of engineers and material sciences looking to the natural world for inspiration for new technologies. Okay, so that's kind of my broad research motivations. So what have I been doing in the last 10 years? So basically a whole bunch of research that at first I pursued because it sounded excited, and looking back on it, I can kind of sort into three main boxes. So a whole bunch of research on how feathers are used for sound production, how feathers are used in flight, and then I'll finish talking about some recent work on how feathers are used to produce color. So here's me. I got my start at the MVC as an undergrad. These were the best old photos I could dig up. So here I am. My first research project was with an MVC grad student studying the Anna's hummingbird. So we would go out to the Albany Bulb and we would record Anna's hummingbirds doing display dives. They do this big elaborate display dive, and at the bottom they make a distinctive chirp. Oh, that's really quiet. And now I've turned it off. They make a really loud chirp at the bottom. And some researchers said that's no big deal, it's a vocalization. Others had put forward that it was a mechanical sound. And so we set about to go and test that. So we'd go out to the Albany Bulb and we recorded hummingbirds making sounds. And then we proceeded to do some experimental field manipulation. So we would remove sets of tail feathers, and we found that if we removed the outermost tail feather, the chirp sound is gone. If we remove other tail feathers, say anything other than the outer ones, they still make the sound. So then, for my undergraduate honor thesis in the MVZ, I did some lab experiments. I took the feathers that we had plucked from the birds and I held them in front of a stream of air. And I found that with sufficiently fast airflows, the feathers will start to vibrate and they whistle. And so here's the outermost feather whistling in an airflow all by itself at the same frequency as the dive sounds. So with that set of field and lab experiments, we were able to show that the Anna's hummingbird is actually singing with its tail. So the chirp that they make at the bottom is sonation or a mechanical sound produced by the feather. So Anna's hummingbird is one of the hummingbirds in the bee hummingbird clade, which is about 35 species that live in North and Central America. Most of those species are known to do a diving display similar to the Anna's. And they are known to have an incredible diversity of feather shapes, right? So given that, it begs the question, are the rest of these relatives with their different shaped feathers making sounds, and how does that feather shape relate to the sound that they're producing? So this launched about 8 years of work on hummingbirds with Chris Clark and some folks at Yale. We proceeded to do a whole bunch of field work. I ended up personally working on 18 species of hummingbirds across 5 countries. To basically document their courtship displays. A lot of them, we knew they were doing displays, but we didn't have any recordings of them or anything. So we would go out, document the displays, and run these experiments, the field experiments, and bring back feathers for the lab experiments. And what we found was, by and large, these bee hummingbirds are producing sounds with their tail feathers. And again, it looks like the different shaped sounds produce different pitches, right? So they're singing at different frequencies thanks to the changes in the feather shapes. So this work was a ton of fun, and it led to, you know, basically the discovery that hummingbirds are singing with their tail feathers. Some further work by my colleagues, you know, went in to really basically come up with a new mechanism of sound production for birds, right? So we had wing snapping, we had stridulation. Looks like these feathers are producing it with what they call aeroelastic flutter. So basically the feathers are fluttering in the wind like a reed in a wind instrument to produce this sound. And along the way, we also got a new species of hummingbird. We did some work in the Bahamas, and we realized that the group, the population that lives in Anagua, the southernmost island of the Bahamas, basically looks nothing like what you find in the rest of the islands. And so we put in a proposal to split it as a new species. So that was sort of the hummingbird work. In a brief nutshell, basically these birds are using their feathers to make sounds, and the differences in shape affect pitch. Okay, so the second big project that I've been working on has been feathers and how they function in flight. This is some more recent work. So, like I said, everyone knows birds fly with their feathers, right? It's like the quintessential function for feathers. And in flying birds, their wing feathers all have basically a very particular asymmetrical shape, right? So in all flying birds, you're going to find that the outer leading edge of the feather is always much thinner than the trailing edge. And we've long been able to appreciate that this asymmetry in the feathers is a pretty important aerodynamic function. In the airflow, it basically helps prevent the feathers from twisting and keeps them, lets them maintain sort of a stable pitch during the airflow. Okay, so asymmetrical feathers, important aerodynamic adaptation. Archaeopteryx also has asymmetrical feathers, right? Our first relative of birds. And more recently, we have turned up many different examples of stem taxa between dinosaurs and modern birds that have things that look like wings, and the feathers in those wings are asymmetrical. So if modern birds fly on asymmetrical feathers, and Mesozoic stem birds have asymmetrical feathers, this of course raises the question, could these stem birds fly, right? I'm going to pause here. We have been arguing about this for well over a century, so I'm going to rephrase the question to something a little bit more manageable. The Mesozoic and the modern birds look like they have the same feathers, but is that really true? Do they actually really have the same form? So they're both asymmetrical, and I pose this question because I did some work looking at how feathers grow into different shapes, and we need to appreciate that feathers are branched, right? So they've got a central shaft, and then they've got these secondary branches called barbs, and it's the barb geometry, right, the length and angle of these branches that determines the shape of the feather. So doing some modeling work about how feathers grow into different shapes, I was able to show that feathers can make the same shape with different barb geometries, right? So you can have two feathers, two asymmetrical feathers, exactly the same shape, but they can have very different underlying branch structures to them. And so I posed the question, do the modern and Mesozoic feathers that got the same shape, do they have the same underlying branch structure? So to get at this, I surveyed modern birds and came up with a sample to just kind of cover the taxonomic diversity as well as the size and flight style diversity. So I've got hummingbirds through albatross here. I needed loose feathers to do the measurements, and so I basically just spent a couple of years preparing skeletons for museums and then saving the feathers that would have otherwise gone in the trash. So I basically made my own feather collection, measured the branch angles and asked, do they vary, and how do they vary with shape? And I found in the modern birds that basically they look to be using branch length, so barb length to get the shape of the feather. That correlates very strongly with width. Barb angle does not correlate strongly with width. So it looks like independent of the shape of the feather, we see different angles within these flight feathers, and we see different angles relative to sort of the function of that feather in the wing. So when a part of the feather is forming the edge of an airfoil, right, the cutting edge here, these red veins, they always have very small barb angles, independent of the feather shape. Trailing veins, in contrast, in purple, always have very large barb angles, right? And so we find there are these differences in the structure of the feather, that underlying branch structure that's independent of the shape of the feather. So there's more than one way to build these feathers. Okay. So we find that generally true of modern birds. What about the Mesozoic birds? So when we look at the Mesozoic birds and we measure their leading and their trailing veins from these fossils, we find that across the board they always have very small angles. So for whatever reason, these Mesozoic stem taxa, despite having asymmetrical feathers, seem to be missing very large trailing vein angles. This guy here, sort of our largest trailing vein angles, that's the enantiornithine. That's the closest stem taxa we had in our sample to crown birds. So if we look at this across the phylogeny, what it looks like is that modern birds have sort of a particular branch structure to their asymmetrical feathers. They've got small angles in the leading vein, big angles in the trailing vein. We see that in sort of the closest relatives, but more basal taxa appear to be lacking this. They just only have these small angles. They don't have this angle asymmetry that we see. Incidentally, so it looks like there's been sort of an evolution in the feathers. We've got a morphological transition through time. We get it about here, crownword of Confucius ornis, and this also happens to be where the alula and the ossified sternal keel show up in the fossil record. And so, kind of taken together, it could be indicative of a major transition in at least flight capability, if not flight itself. So what we've ended up with is that the modern and Mesozoic asymmetrical feathers, they have the same overall shape, but they don't have the same underlying structural form to them. So that begs the question, does it matter, right? What does differences in structure get you in terms of aerodynamic function? And in particular, if modern birds have big angles in their trailing veins and the Mesozoic birds don't, does that impact how these feathers work during flight? And I don't have an answer for you yet because I'm currently working on it. I've started 2 separate collaborations with some mechanical engineers at Stanford and Rolinda Catt, who's an aerospace engineer in England, and we're looking at a couple of different ways these different branch structures could be contributing to the function of the flight feather. So kind of in the middle of that, and maybe if I come back to the MVZ again, I'll be able to let you know just in what ways these feathers are impacting the function. So it's basically TBD. We know that the feathers are different, and it remains to be seen if it actually has a functional impact on flight. Okay, so the last kind of group of research that I've been doing concerns feathers and color, and this is sort of more recent work, so I'll spend a bit more time talking about that. So we will start appreciating this bird's display. So this is a suburb bird of paradise from Papua New Guinea, and he has this very complicated display, which he will get into shortly. He flips feathers up to create a shield. When a female shows up to his perch, he then starts dancing around. He's snapping his wings, and you can see he has turned himself into a blue smiley face. So there's a lot going on with this display. she leaves at the end. So whatever that is, it's not good enough. Okay, so what is going on in this slide, right? We can talk about this display and the behavior in so many different ways. We can talk about the dance he's doing, the mechanical sounds he's making with his wings, the calls he's giving with his voice, these beautiful, brilliant, blue, shiny feathers. Basically what I'm going to end up talking about is the thing that you ignore, which is this incredibly dark black frame that's making the disc. Basically I'm going to present some work on basically how those feathers are so black because they're incredibly black. Here's the paper we just came out with in the last year with our very technical title. The press came up with a much better title. Black holes. So that's what we're going to talk about. How do these birds make such black feathers? So if we look across birds of paradise, we find a lot of different examples of birds with bright, shiny colors and incredibly dark black feathers surrounding those colors. And there are also some close relatives. They're black, but they're just not as black. And this is borne out in the reflectance measurements. So the sort of normal black feathers reflect about 2 to 40%. Very respectable. These super black feathers, which we just look at and they look way blacker, are incredibly black. They are reflecting less than 1% of the light. This was so low that my colleague who had to take the spectrophotometry measurements really struggled to actually get a signal. She mostly just got an error. These feathers are incredibly black. To put them in perspective with some other super black materials out there, we have super black butterfly, which was measured at reflecting 2%. NASA has gone to great lengths to make super black materials to make their space telescopes better. They can get down to 0.5% reflecting, and this is the gold standard, Vanta black, blackest material out there, and it's 0.04% reflecting. Our darkest bird of paradise, Astrapia, comes in just under the Vanta black. And so as far as we can tell, these birds of paradise are basically one of the darkest materials known to man. How do they do this? So the way the other super black materials are working is through a mechanism called structural absorption. So if we consider a flat plate, right, light strikes that flat plate, it's going to reflect off of it, it's going to split into 2 component rays, right? So we're going to have a component in blue here that's going to get transmitted into the material, where if you have melanin, it can get absorbed, and some component of that ray is going to reflect off, right? Now, you can mess with the refractive index and the angle and stuff like that, and you can make sure that you absorb all of the light that gets refracted into the material, but you can only do so much, right? So even if you absorb 100% of that blue arrow, you still will have, you won't have 100% absorbing material because there's always going to be some component that gets reflected off. So folks in optics have long appreciated that to get something to be even darker, if you add superficial microstructures at about twice the wavelength of light, then you can convince the light to bounce off that material more than once, right? And by doing so, with each additional bounce, you'll get a chance to absorb a little bit more of the light. And this is how you can drop that reflectance down to really low. And there's plenty of lab experiments showing that this is work, right? So you can take some sort of material, you can manufacture it as a flat plate. In this case, it reflects at 70%. You take that same material, you manufacture it with these superficial microstructures, and you're down to 15%, right? So it's a pretty effective way to drop the reflectance down. And that's basically what the butterfly is doing, and that mechanism that these man-made materials are doing. So is that what the birds of paradise are doing? If we look at sort of our example of a normal blackbird and a super blackbird, if we look at the microstructure of these feathers, we see that this guy, this is a normal feather, right? This sort of normal standard microstructure for a feather. If we look at the super blackbird, we can see that they're doing something completely different. So they've grossly modified the barbules, the sort of third branches of a feather. Instead of having them lay horizontal in the plane of the feather, like a typical feather and what the normal blackbird is doing, they're sticking the barbules, these branches, up straight up out of the plane of the feather, and they're basically creating a little bottle brush array. right? So these guys much darker, very different thing going on with their feather microstructure. So with that in hand, I said, that's great. We can test this hypothesis, right? So the hypothesis is that this feather down here with all this extra microstructure is scattering the light multiple times in a way that the normal black feather is not, and so they're getting structural absorption. So proceeded to do a bunch of optical simulations and realized after the fact that we had actually already proved our hypothesis because we did CT. We did SEM, and to do SEM we gold-plated the feathers. So when we gold-plated the regular black feather, it turned gold, not surprisingly. When we gold-plated the super black feather, it's still black. So this is pretty good evidence that it's structural absorption. If you can cover it in a shiny metal and it's still black, and way blacker than anything else, it's a pretty good indication that these feathers are not just relying on melanin pigments or internal structure to maintain that super black appearance, right? Okay. We figured that out after the fact. So what I did do was go on to basically test the hypothesis that the super black feathers with their modified structure are scattering the light more than a normal black feather. And to do that, I ran some optical simulations. And so these are ray tracing simulations. It's something used in optics for the development of objects and stuff like that. And so you take something, you specify a 3D geometry. In this case, it's a tube with a bunch of lenses. You can specify the index of refraction and the material properties of those objects. And then you can shine light through that system. So you shine light rays, and then light rays propagate through that system based on optical formulas. So to do this for the super black feathers, I needed to get 3D models of these incredibly small microstructures. So we turned to CT. So we were graciously given permission to sample feathers from birds of paradise. So we would pull a super black feather or a normal black feather from the birds, and then we collected CT scans. We extracted the external surface of the feathers from those CT scans, and then we could bring them into these optical ray tracing programs to see how the light is being scattered by these structures. So feathers are really small and they're not very dense, so you can't scan them in sort of your regular CT scanner that most universities have on hand. We tried. If the scan takes 10 hours, the feather will move in 10 hours and you'll get something blurry. We also weren't quite able to get the resolution we needed. These barbules are small. They're basically the size of individual cells. And so to get kind of what we needed, a good 3D scan at these incredibly small resolutions, we went to Argonne National Labs that has the Advanced Photon Source User Facility. So this is a user facility. You put in a proposal to get access to the facility, but otherwise it's free to use, not free to analyze the data. The data sets are pretty big. But this is basically a kilometer in circumference ring. It's the brightest source of X-rays in the Western Hemisphere. And a whole lot of really cool science happens there. And so they've got beam lines coming off of this ring that give you very nice fancy X-rays. And then lab groups set up different facilities at each of these beam lines so that you can do the experiments that you want to. So we found a beam line that could offer us basically high resolution CT scans at 650 nanometer voxel resolution. And because it's such high energy, we could do the scans in 3 seconds, which means we could get the feather in and out before it bothered to move. And so we could get nice clean scans. So here's an example. This is a pigeon feather that I've steamed in iodine, just to show the setup. So after a couple of years of working out the protocols, we were able to get some really nice usable scans. And so here are some cross-sections from our CT scans. Again, these are individual cells within the feather, right? And then here are the barbules. This is 100 micron resolution, so we've got really great detail. From that, I was able to extract 3D models of the external surface of these feathers. So here are the normal black feathers, and again, this is pretty standard, typical feather morphology. And here are all of our super black feathers. Again, you can see this is basically what we're getting from the CT. When we look at it from the side, you can see that they're sticking these barbules up and making this complex, dense array. So then we bring them into the ray tracing simulations, and those simulations worked as followed. So we've got, a setup, and we define a light source and a detector. And here we modeled the optics of our spectrophotometer so we could sort of recreate the same measurements that we had done on the real feathers. We load in the 3D models of the feathers that we got from the CT scans. Here, just some simple blocks. And then we trace rays. So rays are propagated from the light source. They move through the system. If they hit the feather, they scatter, right? Some of the light will be transmitted into the feather to basically test just the external microstructures and their effects on optics, and to kind of control for any potential differences these feathers have in internal melanin. We assumed that all of the light that gets transmitted into the feather is 100% absorbed. And so this allowed us to control for potential real-world differences in the pigments of these feathers. So the light propagates through, scatters off the feather as many times as it does, and then eventually ends up on the detector. And so with this, we're able to basically, the light starts off with some power, amount of radiant power, and as it propagates through the system, some of it gets absorbed, and so we can record how many times each ray scattered and how much power was absorbed as a result of those scattering events. So here's a screenshot. We propagated 1 million rays, so we can get a nice good sample of them bouncing off of the feather. And if we zoom into any one of those rays there and ask, simply, are they bouncing multiple times off the feather? We see that they are. So here's a close-up of one of our super black feathers, the array of branches, and we can see that as we trace the path of these rays, they are sort of scattering multiple times off of these feathers. If we add up what happens to all 1 million rays for all of the different feathers, we find that the super black feathers, the circles here, are scattering the rays much more than the normal black feathers down here, right? And so we can sort of predict how dark they would be. Again, we've assumed that they have the same absorption ability, so we're basically assuming that they have the same melanin content, and even then, we still see that the super black feathers with the modified structures are much darker. So all of that to confirm what our SEM stub showed us was that the super black feathers are structurally absorbing light. So this is cool for a bunch of different reasons. First off is it gives us a new way to modify the appearance of feathers, right? Previous work has focused on pigmentary colors, melanins and carotenoids inside the feather that give particular color appearances. There's been work on structural colors, which are nanoscale level internal structures, which can create blues and greens by coherent scattering. And then there's been a work on surface roughness, basically nanoscale level superficial structures that can impact how matte or glossy a feather is. And what we found here is basically a new way to modify the appearance of feathers, right? So superficial structures at the micron scale, twice the wavelength of light, can, in addition to all of this other stuff, have profound impacts on the reflectance of these feathers and sort of their dark appearance. Okay, so why do the birds have such dark colors? Right? What is this good for? And we've basically put forward the hypothesis that that bird of paradise is doing the same thing as that velvet, right? Velvet paintings, the colors pop, right? They seem really, really bright. And that's because they take advantage of a bit of an optical illusion, right? Vertebrates, we make judgments on how much ambient light there is in our field of view based off of specular reflections, so the white reflections off of objects, right? If you get rid of those specular reflections by, say, surrounding your Elvis in black velvet, right, we misjudge the amount of ambient light in the system, and we sort of perceive colors to be much brighter than they actually are, right? And so, we submit that the same optical illusion that's happening here is basically what's going on in these birds. Like I said, we only see the super black plumage surrounding colors, right? So this bird's got super black feathers here surrounding the blue. We took a look at the back feathers, not involved in the display. They're regular black. They don't have the structural modifications. They come out at the higher reflectance. And so we would argue that, the same optical illusion, the same reason why that color appears so bright to us is happening as well for the female bird. but that remains to be seen. So I'm going to skip the next bit. The arrays were tilted, right, at a 30-degree angle instead of sticking straight up, and so we did some work showing that there's actually a direction to that signal, right? The feathers actually appear blackest when you look at them from a particular direction and with a particular light. So anyone who's interested in displays and behaviors with directional impacts, we can talk more about that. And I'm going to skip along to the next part of my talk, which is to talk about what I've been doing for the last year, which is not research. So I've spent the last year as a Science and Technology Policy Fellow in the state legislature. So this is a program that is put on by the California Council of Science and Technology. It's basically California's version of the National Academies. And it's a nonprofit, nonpartisan body that was created by the state legislature in the 80s to basically serve as an advisor of science for the government, right? And so in addition to the fellows program that they run, they basically respond to requests for in-depth research reports on topics that are sort of technically complex and politically sensitive. So they put out reports on fracking in the state, underground gas storage, IP policy over the years. And they do this at the request of the legislature or the administration or agencies. So, and they also run a policy fellowship. So every year they take 10 PhD level scientists, and they give us about a month of training, and then they stick us in the state legislature as staff. And we spend a year working on policy, basically. So the idea is to basically give the scientists, us, some experience with policy. For me, it was a great opportunity to see just how science actually is involved in the policy-making process and how that science can best be packaged and put forward so that it actually can be made use of by legislatures. And so we get this wonderful opportunity to experience that firsthand, and the state legislature basically gets access to highly trained folks with technical backgrounds. And the idea here is not so much to put us in there for our specific technical background, right? There were no bills on feathers this year, but with basically all of the training in science, the practice in analyzing complex problems and stuff like that are sort of skills that we can bring to the legislature and help out, right? So it's been an incredible experience. We've basically been doing staff work, analyzing bills, proposing amendments. We've had a lot of professional development. We've had the opportunity to meet with heads and secretaries of various agencies. We sat down and had a conversation with Janet Napolitano, folks that are leading nonprofit organizations and stuff like that, and just get a sense of how they do their job and how they got to where they are. And so that's been really fantastic. And the fellows kind of move on to a bunch of different stuff. Some of them go back into academia. Some of them continue working in the state legislature. They move into the federal government or state agencies, go on to work in nonprofits. We've got a few that have become advocates or lobbyists, and a few that have gone on to national labs. So the 10. I'm year 9, so next year will be year 10 of the fellowship. So they've had just under about 100 fellows go through this program. So for my year, I have spent the year working in the Senate Office of Research, which is a nonpartisan body that exists to provide sort of auxiliary research support to the Senate and their staffers. So senators and committee staff can call us up and say, hey, we're working on a bill, we're working on a particular problem, can you do some more research for us on that? So I've worked on a whole wide range of things. Some of these are quick things that I turn around in a day, and some of them are projects that I've been working on more long term. I've spent the last couple of months working on the kelp in Northern California. Northern California has lost 93% of the kelp in the last couple of years, likely triggered by a massive sea star die-off. So they came to us being, the abalone fishery closed. Why did the abalone fishery close? And what can we do about it, right? And so that's led me into this long report looking at basically these really interesting biological stressors to kelp and what can be done in terms of management. I've also worked done a bit of work for the Senate Natural Resources and Water Committee, and here I was analyzing bills, which is more or less the same process as analyzing a scientific paper for a journal, except that you're in active conversation with the author and the lobbyists and the advocates and everyone else who wants to weigh in about how to, basically amend that bill to get it to where everyone can be happy. And so it's a more kind of back and forth process. So here is my policy portfolio for the year. As you can see, I worked on a whole bunch of different stuff. A lot of things I just didn't even know existed a year ago. So I can tell you a whole lot about regional water boards and the specific problems they're facing policy-wise. And we even got into looking at state-funded research programs. What is the state doing, and what can they do to sort of optimize the output from research? So it's been really fantastic. So fantastic experience. I will say that we are recruiting for the next year. So I've got cards and flyers for anyone in here who has a PhD or will have a PhD before next fall. The applications are open, and it's been really a fantastic experience. Okay, so that is my last slide. I'd be happy to take any questions.
Regarding the optics of the feathers, like the pigmentation and whatnot, as I understand it, birds have 4 sets of color detectors in their eyes versus our 3.
Mm-hmm.
Have you tried simulating, how the birds are perceiving the feathers versus how we perceive the feathers? Because I can imagine that if they're perceiving like a different spectrum, they might look completely different.
Yeah, right. So they can see different colors than we can see, but my understanding is that it's what the super black feathers are doing are changing the relative contrast or brightness, and so that would be the rods instead of the pixels. Right. So yeah, absolutely. The work on that sort of being able to interpret the ambient light and stuff like that has been done in some vertebrates. I don't think it's been done specifically in birds, but that's absolutely where the next work should go into is making sure that they really are seeing that contrast in the same way that we are. Yeah.
Are there aerodynamic consequences of the barbules? So the ones that are super black, does that affect how they fly?
I don't know. It could, right? The feathers that we looked at, these are all contour feathers, right? So we haven't seen anything super black on the wings yet.
Mm-hmm.
But whether or not, there's been some work looking at, bumpy rachises in the wings, and that can have a pretty profound impact on aerodynamic flow, but something smaller than that at the barbule level, how that impacts flow would be
I guess I was
wonderful
just
study.
wondering if there's some sort of trade-off between sexual selection and natural selection on these.
Yeah, if there is, we don't know yet. They're making the feathers in the different way, but they're just kind of sticking them in different directions, so whether or not that has, it's still the same amount of material. Feather development is really complicated, so if there are any costs there, it's not clear where they would be yet. Yeah.
In your work over this past year, how often did you feel that you came up with comfortable compromises on bills and so on? How many times were you sort of give up, this is hopeless?
It was our first year, so they didn't give us too many of those really complicated bills that was above our pay grade. It was interesting, right? It was interesting sort of negotiating with different stakeholders and kind of seeing, really kind of figuring out where they were willing to compromise and where they weren't, and trying to craft the policy. I think as we were doing the fellowship, they kept telling us that you can't break California, right? There's a lot of checks and balances, and they said, the biggest thing you'll probably be able to do is kill a bill before you can get some print. It's just someone's going to come forward with like, hey, I think this is a great idea, and you're like, no, the science does not show that. Please don't do that, right? It's not like that, and that I found to be true. Yeah, in the back.
Are there any other groups besides the bird of paradise that have similar morphology on them?
Yeah, so I skipped over it pretty quick. There's definitely other vertebrates. We've got snakes and butterflies. We do have other birds. So again, at Argonne, we get 72 hours, so we just scanned every feather we could think of, because the scans take about 5 minutes. So we started with the birds of paradise. We've got a lot of other examples of super black birds, super black in birds, and it looks like it's pretty widely distributed. So we're planning to work on that next, basically.
for the arthropods? Is it the same type of absorption that you found?
Yeah, so for the snakes and the butterflies, they're up there, right, 98, 99%. If you look at it, the structures are different, right? And let's see, the bit I skipped over was looking at so here's a snake. It's got these little like monolithic columns on its scales. Here is something that's been manufactured in a lab. It's a bunch of carbon nanotubes, right? And here's the feather. I don't have a scale on this, but I can tell you these are different actually scales. This is actually quite a bit bigger. And you can see that they've got very different shapes. And so I've been talking with sort of material scientists that are looking to manufacture this, and they're really interested when I pointed out these biological examples, because it means that there's like a lot of different ways that you can get to this super black ability, right? A lot of different structures seem to be possible and at different size scales, and so I think the best part about looking at this in terms of inspiring technology is kind of giving them a set of options that they can look at into, right? Some of these are going to be easier to manufacture than others, and so they're, as I'm starting this work, it looks like there's quite a lot of diversity there. Yeah.
I may have missed it, but you sort of left the did dinosaurs fly question. It seems to me you could estimate the body mass in relationship to the feather surface, and some of them almost certainly were flying because there's just no other possible explanation.
Yep, folks have done that. They've looked at wing area, wing shape. There's been a lot of work looking at that central shaft. Was it big and robust enough to really support the weight? So we've looked at these feathers in a whole lot of different ways. We're still arguing about whether or not they could fly. So I'm just kind of trying to provide one other way that we can look at these feathers and kind of get at how they're actually able to function. So yeah, there's a lot of work in there that I passed over.
Any more questions? All right, well, let's thank Teresa for a great talk.